The first question for the homework was a sketch showing 12 non-specific immune defenses with a brief explanation for each. So this person did separate sketches for them. Here's an invader running into the physical barrier of the integument skin, or whatever the outer covering is. Second, lots of animals have mucus, so strands of mucus that they're producing from goblet cells in the skin. Fish, for example, and others. And in fact, a lot of terrestrial animals like us have mucus in their mouths and other places. That traps invaders, and then they're shed with the mucus. In the stomach, in the digestive system, it's an easy place to take in hostile invaders, bacteria, parasites, viruses, and the high acid environment kills a lot of them. Enzymes that are produced in saliva or in the gut can degrade invaders. Some are just the normal enzymes of digestion, but others are enzymes specifically targeted at molecules that are common on invading bacteria or other organisms. There are many animals produce small peptides that are toxic to specific bacteria or even parasites. They are small peptides often that are small enough to be taken in, but the ones that are not broken down by the invader may do something like bind to RNA polymerase or something like that and stop normal function and kill the invader. Many animals produce what are called complement proteins. It's actually a catch-all term for a number of proteins. One major category of them are porins. They insert themselves into the membrane as pores, and then that kills the invaders as they leak out through all of these pores. Another important category are interferons. Interferons are produced by infected cells. So they would be in a vesicle here. Here they are made in a cell that's been infected. There's the infectious organism. Here the green interferons being released. They bind to receptors on other cells, and that causes those cells to make more antiviral defenses. When interferons were first discovered, it was thought that this might be a magic bullet for viral diseases, and sadly it turned out that not to be the case. It helps, but it can't overcome a strong viral infection. Non-specific agglutinins, agglut is to stick together, so agglutinins, these are molecules that will bind at least two ends to a bacterium. So if you have a molecule here that spans these bacteria, they're shown in green, that will bind to both, to at least two different locations on bacteria, and they don't stick to other things that are normal self ones, they'll form clusters of the bacteria. And these clusters will block their access to food, stop growth, and stop them from moving to other locations. In the gut, so here's an animal's gut, here's the head. In the gut, there are normal bacteria, and those can outcompete with invaders. So there's some diseases that are dangerous only when the normal gut flora are no longer there. Phagocytic cells, these are macrophages, for example, they can engulf invaders, and some of them also can secrete poisons that will attack and kill a non-self cell. We'll talk about them, and we talk about more about that when we talk about the macrophages. Then there are, when there is an invasion or damage to cells, histamines are released from mast cells and others that then bind to capillaries and cause them to expand, bring in extra fluid, and the same molecules or others can attract macrophages to that area, and that's part of the inflammatory response that we'll talk again about. This was just another example showing some of these with some brief explanations for some of them. Number 2 is sketches showing inflammation in response to an injury and signals that bring macrophages to the injury site. So here is an injury site, and bacteria have gotten in. They have surface proteins like lipopolysaccharide or other molecules, surface molecules like LPS, lipopolysaccharide, as well as surface proteins. And here is a macrophage binding to one. So here we are blown up. Here is the bacterium, reasonably close to the size it would be, probably still a bit big. The bacterium next to a macrophage would be more like that size. It's binding to an LPS receptor, and that is causing it to be endocytosed into a vesicle. That part of the endocytosis process is to engulf, so the cell will expand around it until it closes it off and pull it into the cell. That whole process leads to a second messenger release. So second messenger, here's our cell releasing second messenger as it digests its bacterial prey. And that is binding to a macrophage that is not currently amoeboid, and that triggers it to become amoeboid. Those same signals also cause nearby capillaries to increase their gaps, so that means they leak more fluid. So fluid accumulates. That's an important part of the inflammatory response, probably creates more space for macrophages to move in, and makes larger distances for bacteria and viruses to have to move to attack a new cell. So macrophages accumulate, and they're moving toward that site of injury and attacking all the bacteria. Here's another example shown a little bit more simply. An injury, the contents of a cell triggering, so this release of messages triggering the macrophage to become amoeboid, and also binding to the capillary, making them more leaky, and here making macrophages become amoeboid. Next one is macrophage function, and showing what happens. So here is a bacterium binding to presumably a lipopolysaccharide receptor. That triggers endocytosis, and the vesicle fuses with a lysosome, and that causes digestion. Whatever can't be recycled is released. Finally, the last video was showing killer T cell development and activation, showing a stem cell here with the different subunits simplified a bit, A, B, and C. We need one of each, in this case A1, B1, C3. That's this one, but here's A3, B2, and C1, and yet another A2, B2, C2. So each of those will have a different shape to its surface receptor, and that shape needs to will bind to a particular structure, an epitope, and if it binds to something on an invading bacteria or viruses, so here is a bacteria or maybe a viral infected cell, here is the surface receptor, an MHC class 2, MHC class 1 surface receptor, here it is, and it's showing a fragment of that bacteria or virus. This thing is binding to things that are inside the cell and presenting them on the surface, showing here's what's inside me, here's what's inside me, here's what's inside me. So if that binds, and we need a second signal from, here's a helper T cell, it also has those variable regions. And if its receptor also binds a fragment, in this case from a specific kind of cell called an antigen-presenting cell with an MHC class 1, here it is, and if that binds, then it'll release cytokines, and our killer T cell to be activated needs two signals. One, it has to bind to an MHC class 1 with a fragment of bacteria, and two, it needs this signal from a helper T. That presumably makes it harder to get an autoimmune disease. You don't have just a rogue killer T cell suddenly attacking anything. There also needs to be a helper T cell that says, hey, there's enough here, this is dangerous, do something. So that triggers multiple rounds of replication. So we get many copies of our A2, B2, C2, and most of them go on to, now activated, bind to any cell that has an MHC class 1 showing our invader, and when they do, they have a variety of attacks that they can make that will kill those cells, including things like reactive oxygen species and a whole bunch of others. Some of those cells stay inactive, dormant, as memory T cells waiting for the next infection.